Hi, hello, welcome to this talk all about SCA35. We have a double feature for you today. We have two speakers. The first speaking, Dr. Luca Marsili, who graduated in medicine and did his residency in neurology at the Sapienza University of Rome, Italy, where he also achieved his PhD in clinical neurosciences. He currently works as an assistant professor of neurology at the University of Cincinnati in Ohio. His research activity is related to neurodegenerative diseases and in more detail to ataxia, Parkinson's disease, atypical Parkinsonian syndromes, and other movement disorders, with a particular interest in investigating the genetic, clinical, and neurophysiological aspects of these conditions. We also have with us today Dr. Manuela Basso, who graduated in medical biotechnology at the University of Torino, Italy, and she graduated with a life science degree from the Open University in the United Kingdom and the Mario Negri Institute for Pharmacological Research in Milan. She then moved to the Burke Neurological Institute, Weill Medical College, Cornell University for her postdoctoral training. In 2012, Dr. Basso became an instructor at Cornell and in 2013 was appointed assistant professor at the University of Trento in Italy. Her research activity is related to neurodegenerative diseases and more specifically to ALS, Kennedy's disease. She studies the biochemical alterations of mutations in transglutaminase 6, highlighting the tendency of these mutations to induce a loss of function. So as you can tell, we have two powerhouses in the house with us today. With that, I turn the floor over to Dr. Marsili and Dr. Basso. Welcome. Thank you for joining us.
Thank you.
Thank you, Celeste. Thank you to the National Ataxia Foundation for your kind introduction and the invitation today. When I was asked to present, to give a talk on SCA, I deliberately choose the SCA35. That is probably one of the less known among the SCAs, this huge group of spinocerebellar ataxias. And this led to me to a few years ago when we described a peculiar case, and I will present briefly, of a patient in Cincinnati with SCA35. And then while looking for some basic science experiments on the role of that specific protein, we came through Dr. Manuela Basso, and we realized that she's one of the top-notch scientists on the topic. And so I was so happy that she accepted today to be part of this talk. There will be a shared talk together. So I will be starting with the clinical aspects of SCA35, and then Dr. Basso will guide us through the basic science, the role of this important protein, the transglutaminase 6. So let's start. How was this SCA discovered? This is the original article published on Brain, one of the most prestigious neuroscience journals, in 2010 by Wang and colleague, when they identified a specific mutation in the transglutaminase 6 gene, the TGM6 gene, using a particular technique that is called whole exome sequencing. That is, at that time, was one of the most sophisticated genetic techniques, basically scanning and analyzing the whole genes in the DNA. So a huge study. And they found this specific mutation in a huge family, like 4-generation family of a Chinese Han descent. And they found actually 9 individuals with this spinocerebellar ataxia with a variable age at onset, but the mean age was around 43 years. Now, let's talk about a little bit of the mechanism, who is at risk and how this is transmitted. The transmission, this is the SCA35, the TGM6 protein. And the transmission is autosomal dominant. That means that, as you may or may not know, each individual has 2 sets of gene for each gene of the body. And these are transmitted in an autosomal dominant fashion. So that means that if one of the 2 individuals is affected, 50% of the descent can have the mutation. This gene, the TGM6, codes for this protein that is the transglutaminase 6, as you may have heard earlier. And mutation in this protein may cause some instability in these transglutaminase, in this enzyme activity. And then Dr. Basso will show us what does it mean. Mostly there are a lot of debates, but it's thought that the instability of this protein may contribute to the neurodegeneration that we see in SCA35. So how is this condition diagnosed? Again, this is one of the original contribution. Now we have many more mutations, but this is to show you how mutations in this condition are different from the traditional spinocerebellar ataxias. So the most common one, or the very first one, which were described, as you can see in the bottom of my presentation, SCA 1, 2, 3, 6, 7, 8, 12, 17, and so on. They are all repeat expansion disorder. This means that there is a repeat sequence in the DNA, usually a CAG sequence, that is causing excess of amino acid in the protein. And that means the more are the repeat expansion, the earlier the disease develops. And this is valid for, I would say, most of the SCAs. Differently, in the SCA35, this is a non-repeat expansion disease. So this is a single mutation that could be a missense or a truncating deletion causing mutation. That means that the protein misfunction or doesn't function. And the best test to diagnose this condition is the whole exome sequencing, as we were discussing, like scanning the whole codifying regions, the all genes in our DNA. Let's talk about the epidemiology. So epidemiology of SCA, this is a very rare disorder. Among 5 to 7 cases per 100,000 people. However, for some reasons, Eastern Asian countries, Japan, show an increased incidence of this condition, not only of SCA35, but of many multiple SCAs, including other rare similar neurodegenerative conditions. And SCA35 has been originally described in a Chinese family and is more frequent, I would say, in Asian population, but it's not only seen there. It can be seen also in other ethnicities. Regarding the clinical aspects of SCA35, the symptoms of this condition, as for many other SCAs, this is cerebellar ataxia. So it means that there is an involvement of the cerebellum and the spinal cord, which are somewhat causing lack of coordination balance that is called ataxia, plus some other features. Usually SCA35 is slowly progressive, but we would see there are also cases that may be faster progressors. The age at onset is highly variable. It may start around 9 to 12 years old till the 5th decade, and we saw with the mean age around 43, mean age at onset. It may or may not be associated with cognitive impairment. And at the brain MRI, we see a lot of cerebellar involvement, so the cerebellum is shrinking, and there may be usually not involved the cerebral hemispheres or the brainstem as compared with other types of SCAs. So I wanted to show you this slide. This is the case we described a few years ago together with Dr. Basso, who was a patient we saw that had a different phenotype, not the classical spinocerebellar ataxia, but this patient was having some dystonia, tremors, and later on developed supranuclear gaze palsy. This is a quite distinctive sign of a condition that we call progressive supranuclear palsy, that is a Parkinsonian syndrome, an atypical Parkinsonian syndrome. And fortunately, this patient died, but the patient decided to devote the brain to research. And I think this was very important. We will be always thankful to this decision because we were able to find a specific condition that is kind of proving the diagnosis of progressive supranuclear palsy. That is the presence of these 4 repeat tau proteins. And this is, in the beginning, was a little bit counterintuitive because we have transglutaminase mutation causing a tauopathy. So these are 2 extremely different conditions. But we will then see also with Dr. Basso that transglutaminase may have a role in stabilizing the tau. They may act on the microtubules that are important components of the cytoskeleton of the cells. But the other interesting part, at that time, it was 2020, we kind of did a systematic reviews of all cases of SCA35 reported in the literature. At that time were about 66 cases. And interestingly, as you can see here, 50% in blue were showing prominent ataxia, as you would expect in spinocerebellar ataxia. And only a big chunk of them, almost 40% here in red, were showing other features, and in this case, Parkinsonian predominant. So kind of in line with our case described. This is a complex slide, but just to show you, whenever you see the supranuclear gaze palsy, you should think of different conditions. The most frequent one is progressive supranuclear palsy that is usually happening in patients older than 40 with or without cognitive impairment. If the patient is younger, you should always think to Niemann-Pick type C disease. That is another genetic condition, usually developing earlier in life and with some associated liver and spleen involvement. If there is an abnormal MRI, in this case, I would say the cerebellum involved, you should also think of some SCAs like 2 and 3, but also in this case, this was the first description of SCA35. So symptoms-wise, we described the slow onset, the cerebellar sign that may happen in SCAs. However, it's interesting because these transglutaminases are a huge group of proteins. There are different type of transglutaminase, and they all catalyze the reaction on glutamine. So gluten proteins are rich in wheat and other cereals, and they are probably potential substrates for these transglutaminases. So someone in the past has postulated that there is a relationship between celiac disease, that is the gluten resistance that we know is much more common, and SCA35. There could be a common shared mechanism in causing ataxia in these cases. As a matter of fact, celiac disease may be associated with a type of ataxia, very common. This is an interesting case because when we were studying and presenting our original case presentation, we came across this study. This is a patient manifesting only with some imbalance and tremor, pure tremor. This was reported in the literature. This patient, as you can see, has a bat wing tremor, pretty disabling tremor. And this patient was treated with deep brain stimulation, surgical technique that may be used to treat tremor as well as Parkinson's disease symptoms. In this case, it was used to treat tremors. And the patient improved significantly in his tremor symptoms after the deep brain stimulation surgery. So this is to show you how wide could be the presentation of this condition. And so going to the treatment part, as we were discussing, the postulated relationship between SCA transglutaminases and celiac disease has led to do some trials in a gluten-free diet that is the treatment for celiac disease. Now, probably SCA35 is so rare that it's hard to create like a randomized placebo controlled clinical trial with that. But some cases have been reported successfully improving after gluten-free diet in SCA35. And then as we saw, there may be selected cases responding to deep brain stimulation surgery for tremor. Levodopa is the cornerstone of treatment for Parkinson's disease and could be tried in patients with prominent Parkinsonian features with SCA35. As we saw, about 40% of described cases have some degree of Parkinsonism, so it's not unreasonable to try levodopa. And then for sure, the mainstay of spinocerebellar ataxia is physical therapy for gait and balance disorders. And with that, I will go and leave the talk to Dr. Basso, and she will describe the current state of research in SCA35. Thank you, Dr.
Thank you, Luca. Thank you also because you gave a beautiful overview. I'm not sure I will cover all the points in my slides, but I'll try to mention at possible links while we see the different figures. So if you can go to the next slide. Today I'm going to tell you a little bit more about what happens into the cells and what we were able to understand, even if we are just at the beginning of it with our studies. And I'll also try to mention two other studies, even if I have to say that there are not many researchers working on transglutaminase 6, and it would be beautiful really if the critical mass could in some way increase over time to understand better with different approaches really which is the role of this enzyme, of this protein in the disease. So the transglutaminase 6, the gene is characterized by several exons, and as Professor Marsili just said, Dr. Marsili just said, usually the best way to understand if there is the possibility that SCA35 is present is really to go through the exon sequencing. But this gene encodes for a protein called transglutaminase 6. I think there are animation, Luca, so if you can just go ahead. I don't know, okay. Encode for a protein that is mostly expressed in the brain. So transglutaminases are a big family of enzymes. There are several isoforms from 1 to 7. And I would say that transglutaminase 1 is, for example, the most important for our skin. Defects in transglutaminase 1 leads to individuals, people that cannot correct form the skin. Just to make you understand that in the same family of enzymes, some mutations are really not compatible with life. They can lead to disease as well, but in other parts of the body. And for example, transglutaminase 2 is expressed more or less everywhere and is the most widely studied isoform. Transglutaminase 6 is expressed in the brain, and here where you can see the bluish color, the dark blue color, it's where it is mostly expressed. So you can see that, for example, in the cerebellum here, there is a darker sign, and that is where you have the expression, and also in other parts, in the hippocampus, for example. And what we did, it was just to verify that this is from a study that was published in 2013, just a few years after the mutation in the SCA35 was actually described. And then we tried with the different antibodies to prove that also in the mouse models that we had in the lab, we could see the same expression. And as you can see here, these are Purkinje cells that are the cells that degenerate the most vulnerable one in spinocerebellar ataxias, and this is the expression of transglutaminase 6 that is mostly really in the soma of these cells, so in the cellular part of the neurons. So if we go to the next slide, we can see, let me see, what these enzyme does. So this is a protein that has a specific function in the cell. The only problem is that we don't know exactly why, when there are specific mutations, these could lead to the death of Purkinje cells and manifestation of spinocerebellar ataxia. So this is just a general schematics on the left of the function of transglutaminases, so the entire family of enzymes, and they all do the same. When there are changes in calcium level inside a cell, and that is typical, for example, of a condition of stress, what it happens is that transglutaminases can cross-link, they can bind together different proteins, and that is a sign of damage. And sometimes, though, this function can also be fundamental, for example, to stabilize certain structures in the cells, like, for example, the ones that are important for the cytoskeletal stability. What we tried to do, it was to reproduce in the laboratory some of the mutations that were described. So in a technical term, we clone these mutations in specific vectors. And then in order to understand whether this mutation would augment and exacerbate the transglutaminase activity, this transaminase activity that I described on the left, we put together the normal protein, so the wild-type protein that healthy individuals have, and the one with the mutations that were described with the patients. And as you can see on the right part, whenever you see a black horizontal lines, it means that the enzyme is working correctly. But when you put together the wild-type form with the mutant form, here we took one of them that is, for example, R11C. So it's a specific mutation that was described in one patient. You can see that the color becomes fades, and so it becomes fade and lighter. And that really indicates that the function of the protein is in some way lost. So even if this disease is autosomal dominant, that it means that it's enough to have the mutation in one allele of our body, it seems that when the mutant form is expressed, it can also shut down the activity of the wild-type form. The only big problem that we have in biology is that we are not yet at the point in which we know which is the exact function that is necessary for the Purkinje cells to be healthy. So that means that when the function is lost, that can lead to the degeneration of the neurons. So if we go to the next slide, if I remember correctly, I summarized. This is a very small table. I apologize for that, but it was just to show you a paper that was published by the Difonso group in 2020, in which they collected the mutation that were associated to transglutaminase 6 and Spinocerebellar Ataxia 35. So we are back to 5 years ago. And I wanted you to notice 2 points in this table. The circles or ovals are just showing that the numbers of patients that have been described so far and for which the transglutaminase 6 activity was tested are very low. So one of the things that we could aim is really for more people to test the activity of this enzyme in order to understand if the function is normal or not, and which kind of mutation is really associated to the patient. Because then instead in the square part, you can see I highlighted how many of all the mutations that were described showed a reduced activity in vitro, and that was done by different labs around the world, not only from my lab. So really suggesting that in most cases, mutations in transglutaminase 6 that have been associated to SCA35 also have a reduced activity in the laboratory. And that really build the case for a loss of function of this enzyme, or a dominant negative loss of function from a biological point of view. So if we can move to the next slide. Yes, it's the reduced activity. I want to show you two things that we prove in order to really make sure that some of these mutations would have a detrimental effect if expressed in neurons. Because you will see later, SCA35 is extremely rare, also as Dr. Marsili said. So there are also doubts on the fact that really transglutaminase 6 is really the gene that is mutated and causing the disease. So in the laboratory, in order to see with our eyes if mutations that have been described would have a toxic effect, what we did was to overexpress transglutaminase 6 in the presence of the mutation or in the normal wild-type form. And as you can see, between the gray and the black bar, there is a reduction of the number of neurons that here we counted by looking at the red color. MAP2 is a cytoskeletal marker of neurons, and the GFP instead are the neurons that express transglutaminase 6 wild type or mutant. And we counted how many neurons survived over time in the presence of the mutation or not. And as you can see, we counted less neurons in the black bar compared to the gray bar, really showing that the mutation, at least this kind of mutation, looks detrimental in neurons. It's damaging neurons. So if we go to the next slide, we did the same experiment, not in vitro, so in cells that we plated, in neurons that we plated, but in vivo. And we used the Drosophila in collaboration with Uday Pandey at the University of Pittsburgh. And again, exactly what, just to really confirm what Dr. Marsili said earlier, you can see that in the black bar on the left, there is a reduction of this protein. So the protein is less stable. And the flies that are red, they show us a shorter lifespan compared to the gray one that are the one expressing the wild-type isoform. And so we also proved that in vivo, this mutation gives toxicity. But again, we looked at the general survival of the flies in this case, okay? And but really to make sure that the loss of this enzyme, it's bad, it's something that it's detrimental, we had to go to a more complex organism, that is the mouse, because in the mouse there is the cerebellum, and we wanted to understand really if the loss of function of transglutaminase 6 could really be associated to alteration in the movement and in the coordination of the movements. And so if you look up, please go to the next slide. You can see that what we did, thanks to the support of a seed funding from the National Ataxia Foundation a few years back, we revitalized a mouse model that was produced by the European Mouse Mutant Archive. So we asked this consortium to revitalize this mouse model that has, from a technical point of view, something that blocks the normal production of transglutaminase 6. So they inserted inside the gene a cassette, the one that is written LAC, Z, and NEO, that really makes the entire protein, the entire mRNA, messenger RNA and protein less stable. And once we revitalize these mice, we characterize first the reduction in the protein level and mRNA and messenger RNA level, and then also the behavior of the mice, because we really wanted to understand if we can find a model to study better SCA35 and eventually also look for therapy or understand better the function. So if we go to the next slide, these data are actually not published yet, but we have the manuscript submitted, in which you can see that in this model, we can see a reduction of the messenger RNA level. The RNA then gives protein, and we also proved that at the level of the cerebellum, the expression of transglutaminase 6 is reduced in the mouse where is written KO, knockout, okay? So this is the mouse in which it's not a real knockout because we simply use the strategy of this cassette instead of crossing them in another system with some different mice. But that is enough to reduce the protein production. So said that, if we go to the next slide, we started to look at the mice over time. We, for example, monitored their body weight, because many times if the body weight is reduced, that is typical of some neurodegenerative diseases, like, for example, amyotrophic lateral sclerosis, Huntington disease, these diseases that are really fast progressive and they take really to the loss of the body mass. But in this case, we didn't see much of a difference, nor in males or in females. If we go to the next slide, we decided then to look, and also the animation, thank you, Luca. We decided to monitor the mice up to 16 months. So, and we tested the mice at we were looking at them every week, but we tested them at 1, 4, 7, 10, 13, and 16 months. We decided to perform the classical tests that are usually done to understand whether there is ataxia, if there is a problem at the cerebellum. And we also, either in the coordination with the Rotarod test, because we use a velocity that can be maintained by the mice if they can coordinate well their movement. And then we use a ladder test in which there are the different ladders have a different distance one from the other. So again, only if the mouse has a good coordination can move through the ladder without making mistakes, without having a misstep, for example. And then we also did the balance beam, where the mice can walk on a very tiny 8 millimeter bar. And if they have a good equilibrium, they can go through it. But if they have an ataxic phenotype, they will have some problems. So now I'll show you something that we still haven't understood well, and there will be the need really to do more work on that. So Luca, if you can move to the next slide. You can see we started with here I'm starting presenting the balance beam, and we started to look at the male. And as you can see, at 10 months, in the balance beam, where they need to walk in a very small space, in a very tiny bar, at 10 months, the knockout mice, they start to have to make many more mistakes compared to the other mice. But then when they become older, the mice usually they also become bigger, and it's more difficult for them. So we see the difference only at 10 months. In the next slide, though, you can see that the females do not have any problem. And that was surprising, because we tested the reduction of the protein both in males and females, but only males had that. So if we go to the next slide, we look also at the ladder test. As I said, we counted how many times the mice made a misstep. And as you can see here, at 16 months, so towards the end, probably I inverted that, but at the end of everything, there is an increase that is significant, almost significant anyway. So the mice really make many more mistakes, the one in the dark gray bar, compared to the wild type. So there is something also here that is not working very well for these mice, but it shows up at the end of the disease. If we go to the next slide, though, again, females do not show, it's 7 months. At 7 months, we see this defect, but the females do not show any of these defects. And that it's again puzzling, because we can reproduce the alteration of coordination in males, but not in females. And we don't know why, because they were from the same cohorts. And then we went ahead with the last test, that is the rotor test, that was done with a constant speed. That it means that really you don't test for the muscle resistance, but only for their coordination ability. So the mice were walking in a very slow rod, always constant. And as you can see here, the male mice, starting at 10 months and then up to the 16 months, show in the knockout form really a reduced ability to walk into the road, so showing a defect in the motor coordination. But in the next slide, we can see that the females did not have any problem. So we are now at this point in which it would be nice to understand why we notice this important sex bias. Maybe this is just something that is related to the mouse model, and that we cannot really reproduce in the laboratory. Or maybe it can also tell us something more about a higher risk for males to sense defects in transglutaminase 6. But this is something that would require many more studies from many more researchers, in my opinion. If we go to the next slides, I really want just to point out that, like in many other ataxia form, we didn't see an increase of neurofilaments. I'm working on amyotrophic lateral sclerosis, and neurofilaments are really the gold standard as biomarkers, and they are pieces of the axons that are just released in the blood. When we measured that in these mice, we didn't see anything significant. So really suggesting that neurofilaments are not, that if there is any defect, this is probably really central and not peripheral, like in ALS, not at the level of the motor neuron and muscle neuromuscular junction. So if we go to the next slide, I'm really closing up and leaving space for the question. These are the last slides. And if you go through all of them, it's perfect, Luca. So here, I just want to really wrap up by saying that from the evidence that we have and that other labs that publish on transglutaminase 6 independently have or showed, it seems that really there is a reduced activity of transglutaminase 6 that then leads to this alteration in the coordination. But what we still miss is understanding the real function. We will try to analyze the tissue that we collected from this mice model, or it would be beautiful to make possibly even iPS-derived organoids, cerebella, to really study better the function. But we still have this open question of what is the function of this protein. And then I wanted to mention about some controversy that were discussed in the scientific community about the fact that that there are some some genetic analysis suggesting the fact that at least in the Asian population, there are many individuals with variants in transglutaminase 6. And the variants in transglutaminase 6 have been also been associated to Parkinson's disease. But as Dr. Marsili showed you, there could be also Parkinsonism involved in the Spinocerebellar Ataxia 35, and this is what they observed. So there are some studies really showing that the variants in transglutaminase 6 can be found also in other neurodegenerative diseases, but also in healthy controls. Though what I want to say is that the variants that were described in this paper that I put in the blue box were not really characterized from a biochemical point of view. So we don't know if these variants really lead to a loss of function of the protein. We saw some toxicity, we saw some enhanced toxicity by some of the mutation, and I think that anyway there is still more, and we can go to the next slide, that we can do and study all together to understand better. Because if really the loss of transglutaminase it's important, then there are so many technologies nowadays to try to replace the protein function genetic, from a molecular biology point of view. So other evidence could help to reinforce what we and others observed. With this, I think I concluded my slides, and I give space to Celeste. Thank you very much.
Wonderful. Thank you both so much for this presentation. We're going to switch into the Q&A session now. The first is from an attendee who wants to, if you could explain again about the different presentations of SCA35 in terms of Parkinsonianism versus ataxia, in terms of the different kinds of symptoms that people could be experiencing. Could you clarify about this again?
Yeah, sure. I think I can take this one. Yes, this is really fascinating because, as I mentioned in my presentation, spinocerebellar ataxia means mostly ataxia, lack of coordination, imbalance. However, when reading that's why we did a huge search in the literature. While reading all the cases reported so far, and this was up to 2020, there were many with mixed phenotype. This doesn't mean that the patient doesn't have ataxia, but they may have prominent other features. That is what we call Parkinsonism, so mainly slowness of movement with associated different variable mixed features like tremor, rigidity, stiffness in muscles, postural instability. And this gave rise to this questioning: is this a specific condition, or is just the association of the two, like a patient with Parkinsonism that gets tested and found to be harboring this variant in the DNA? In the case we described, that was one of the questions of the referees of the application. They said it may be that this is just, a coincidence. However, in our case, the patient had pathology-proven PSP, so progressive supranuclear palsy. That is, something we cannot question, and it is a very rare condition, right? Plus this mutation, that is a very rare mutation. So putting these two super rare conditions will have been like an extra super rare variant of something, like having two different diseases is really unusual and rare.
Definitely.
So that's why I would say, just to conclude this is a very important question, and that's why we need more cases to be studied, diagnosed. And in our case, having the pathology, I think is one of the few cases with pathology proven that I'm aware of. So it's very important to study the biology and the pathology to get aware of what's happening in the brain. But that's a very important question. Thank you.
Of course. Thank you, Dr. Marsili. And we've had a few people submit questions that are not related to SCA35. If you do have questions about other forms of ataxia, please email them to research@ataxia.org. We're happy to help you to direct you to other resources. All right, moving on to our next audience question. We have an attendee who has shared that they have tested positive for the gene mutation associated with SCA35, but their doctors are hesitant to give them the formal diagnosis because their symptoms fluctuate week to week. They have good weeks and bad weeks. Do you know of any cases where SCA35 comes in waves like this, where there's fluctuating symptoms?
Yeah, not to my knowledge. However, I would say again, from what we read in the literature, many of these cases have just been, case series reported to be tested positive for. So this doesn't mean that the gene has been tested in a lab, as Dr. Basso was saying. So we cannot the function of this variant. So whenever we found a mutation, we can say this is pathogenic, likely pathogenic, or a variant of unknown significance. So I will go first and try to understand, is this a pathogenic mutation or not?
I see.
And then based on the variant, we can tell with a certain degree of approximation if these symptoms may be associated or not. I can tell you there are cases with a very rapid and quick progression that are quite, severe. Other cases are very mild. So it wouldn't surprise me if in the beginning there is some degrees of fluctuation and then it becomes more like a stable condition. However, I think this is also raising an important point. In the genetics clinic, it's important to have a genetic counselor to address all these aspects together with the clinician and try to guide patients through these diagnoses, because sometimes it's really hard, even for non-specialized clinicians who may order the test, but they don't know how to address all the questions that may raise before.
Of course. Thank you so much for clarifying that. We've had a few questions around gluten-free diets. So if you could elaborate again, do you recommend, or is it recommended that people with SCA35 try a gluten-free diet? And could this be beneficial for their symptoms?
I think, as I mentioned, the disease is really rare. So we don't have room for double-blinded, placebo-controlled clinical trials. That would be the best way to address the question, like giving, patients, probably the same patient at different time points in the study, either placebo or the gluten-free diet, and see how the symptoms may change over time. This is really hard to do. However, in single, case, I've been told that patient had started the gluten-free diet with some benefit. I think it's not harming, so it's worth to be tried. I think there is a huge possible placebo component here. As we may see, I was, just talking with my colleagues about a trial on PSP. This is a different condition, but that has been stopped due to the probably placebo effect that was mimicking some response, but then they had to stop it because they realized, looking at the data, there was not really effectiveness of the drug. So it's really hard to answer this question, but I would say in the uncertainty, I think it doesn't harm. I don't know, Manuela, if you want to add something on that, if you know of other cases that may have been reported.
I think that gluten can give inflammation, no? And so everything that gives inflammation also for neurodegenerative condition in some way could exacerbate some of the problems. So for now, I know that in UK there are clinics where the gluten-free diet is really advised, much advised for patients with SCA35. And I also know that there are some works showing that autoantibodies against transglutaminase 6 specifically are observed in patients, in the serum of patients. So again, for example, I've been in a gluten-free diet for other reasons for a few years in my life. I think it's not maybe so easy, but it's also not so difficult. So as you said, maybe the best thing is to try and to see if there is any beneficial effect.
And if anyone on this call has questions around gluten-free diets and ataxia, we do have a webinar about gluten ataxia specifically, where we have one of the experts in this field answer questions around gluten-free diets specifically. So that's on our YouTube page. I'll plug that resource.
It's the best, yeah.
Moving on to another question from an attendee who shares that his son-in-law was recently diagnosed with SCA35, that his son-in-law's father likely also had ataxia but has passed away. What kinds of questions should they be asking their neurologist to learn more about this condition? And could you just quickly go over again any interventions that are available for people with SCA35?
Yeah, no, I think it should be really based, as I tried to summarize in my presentation, case to case. So what is the prominent symptom? What is the most disabling component? And then go through with, the neurologist is the specialist who may guide you through how to address the different symptoms. So we know that, for example, some cases have been reported with tremor responding to surgery. And so if the tremor is disabling and if there is no response to medication, we never start with surgery. We start with medications. And if it does not respond, then we can consider the surgery, of course, having on board a huge team of different specialists on board discussing pros and cons of the procedures. I would say physical therapy is very important if there is ataxia imbalance. Exercise is the best way to counteract and slow down any neurodegenerative disease, also Alzheimer's disease. So memory problems may be slowed down by physical exercise, not just being mentally active, but really physical activity is the way to fight this. Of course, within the limit of what is possible. Each patient has a different baseline, has a different performance status that may be associated other, cardiovascular pathology. So we don't want to push too much, right? It case by case. But I think the neurologist is really the right specialist to ask and to start, kind of a process with them and to be guided on what's the best way to get your brain healthier.
Definitely. Moving on to another question from an attendee about some of the mice study data that you were sharing, Dr. Basso. She just shared that the mice study data was interesting. So she's female, but her son seems to be progressing much faster for ataxia than she was at her age. Could you elaborate again on maybe some differences you may see in progression rates in the mouse models you're studying, or if Dr. Marsili, in terms of if there's anything known in humans? I know there's very few human-based studies out there, though.
Yeah, from what I want to say, the only thing I can say is that in the lab, really, the data is what the data is, and this is what we saw. So I think there is room for more investigation on that, because I think it's extremely, anyway, the sex bias and the sexual dimorphism in the neurodegenerative field is also becoming relevant. And I think that in this case, really the mice are telling us that for some reason that we don't know, males, when they exercise, because they were tested every year and then, they just develop symptoms and the females do not. And we see the same also in a model of amyotrophic lateral sclerosis. I don't know why, but also in that disease there is a sex bias. Maybe the changes in this enzyme are just altering something related to the cells that are involved in motor coordination or in the Purkinje. I don't know. I think we should study it better, honestly. But it's something that I would keep in mind.
More research needed in terms of trying to figure out, these are big questions and they take time to answer. So thank you for your part in helping us give us some answers over time. We have another question about your slides, Dr. Basso. There was, we have an attendee who asks that on your slides you were making, saying that the genetics was complicated, that there's sometimes not very clear genetic connections.
Mhm.
Is SCA35 related at all to ataxia with unknown ataxia or without family history? Is there any connections here, or could potentially people be misdiagnosed?
Oh, yes, this is what really somebody is starting to say. There is the possibility, for example, that transglutaminase 6 variants would be a risk factor and not a causative factor. And that's the reason why we need, in my opinion, to study the function better. In any case, it would be useful for both cases, but it could be that maybe in an unknown genetic situation, specific variants of transglutaminase 6 can enhance or prone neurons to degenerate first. So this is I wanted to mention that just because it doesn't mean that having a variant in transglutaminase 6 it's for sure Spinocerebellar Ataxia 35. It's for sure is a form of ataxia, form of alteration in the motor behavior, but it may not be only may not be only one disease, or maybe like one specific disease. It may be just a risk factor. So it's something that, in my opinion, needs to be studied better. I don't know if I answered, but
No, that's perfect. And that actually leads well into our last question here. If people, because we've mentioned that more research needs to be done in order to find answers for us. If people on this call want to get involved with SCA35 research, what recommendations do you have for them to get started? Should they be contacting their
if you need
neurologist?
tools, from my side, from the biological point of view, I have the tools and I'm happy to share and to discuss and to plan experiments, whatever. And I think it would be very important to do that, honestly.
Wonderful. And then for patients who want to get involved, maybe donate samples, things like that. What's the best way to get connected?
Oh, I think in terms of getting, for example, blood to make IPS or organoids, I think the best way is just maybe a direct contact with the neurologist, and then through the ethical committee, then go through all the process of making those, okay? Keeping all the rights to the patients, of
Of
course,
course.
whatever they donate. Yes. And then if anybody wants to drop an email or have a chat anytime.
And if you need any assistance reaching our speakers today, please feel free to reach out to research@ataxia.org. We're happy to pass along messages. But it sounds like a good first step is if you already have a neurologist, chatting with them and letting them know that you are interested in research and you'd like to learn about these opportunities.
Yes.
Well, wonderful. We are at time. So Dr. Marsili, Dr. Basso, thank you so much for joining us today. We
Yeah.
really appreciate your time and expertise teaching us more about SCA35.
Thank you, everybody.
And to everyone on this call, I hope that you enjoyed the past hour with us and learned something new about SCA35. And wherever you are in the world, good morning, good afternoon, good evening. Have a wonderful rest of your day. Take care, folks.